Hi everyone, this is lesson 4, antibodies and response to infection. So this is part of the AQA A-level biology section 3.2.4, cell recognition and immunity. So the part of the spec we are going to look at in this video will be related to the definition of the antibody, looking at antibody structure, and then how they destroy bacterial cells and also viruses, and then we'll be looking at what we call the primary and secondary immune response. Looking at the different immune responses, we know that the immune response related to B cells is when they produce the specific antibodies, but we really need to know what is an antibody. Well, it is a protein, so it's a protein made of 4 polypeptide chains, and it is joined together by particular bonds, for example disulfide bridges. Each antibody has 2 specific binding sites in the variable region. So we're looking at this part here and this part here. They're known as antigen binding sites. So these are complementary shape to a specific antigen to form what we call an antigen-antibody complex. And each binding site consists of a sequence of amino acids that forms a specific 3D tertiary shape. So there are 2 main ways that antibodies will destroy antigens. The first one is what we call agglutination. So this diagram here shows agglutination, and as the antigens, they bind to 2 parts of the antibody, to the variable site, then they get clumped together, and that means that it makes it easier for these guys here, the phagocytes, to locate them, and so they're very close together. So that stimulates phagocytosis, and that engulfs the cells to which they are attached. So once antibodies are produced, there are 2 responses that may occur. So the first response, as we can see in this graph, is the primary response. And we can see the first infection at day 0, and you can see the concentration of antibodies increasing, and it needs to get to a certain level where immunity is required. And as you can see, there is a delay. So there's the first delay is between the first infection and actually antibodies being produced. So we're going to talk about that first response, and then the second thing is the secondary response. So once we've actually been infected or we've had a vaccine, then we have antibodies circulating, we have memory cells circulating in our blood, so that keeps antibody levels at a particular level. And you'll notice here that second infection, the response is much quicker, it's much faster, and it increases many more antibodies much more quickly. So we're going to talk about both of those responses. So the primary response. So first of all, the immune system is activated when the antigen enters for the first time. However, this is slow. The immune system is slow as there aren't many B cells that can make the correct antibody. So eventually the specific immune response will develop enough antibodies, but the infected person will show symptoms of the disease. After exposure, both T and B cells will then produce what we call memory cells, and these remain in the body for a long time, and these remember the specific antigen that they have come across before. In the secondary response, if the same pathogen enters the immune system, it will produce a quicker and stronger response. So what is happening is that clonal selection happens faster, and B cells are activated and they divide by mitosis much more quickly than in the primary response. And it produces a large number of the correct antibodies, so reaching a required level for immunity much faster. Hope you found all of that useful. Please do subscribe to DrBiology. My next video in this unit will be looking at monoclonal antibodies. Hope to see you soon.